like
we are on live, sir.
Yes.
Good morning, one and all. So, guest speaker, sir, Dr. Srinivas Katta, our director, Dr. Kumar, sir, principal, Dr. K. Prasad, all the dignitaries. Welcome back to the 5th day session of FDP on drug discovery and development. So, before going to the session, I would like to introduce our director, sir. Our director, Dr. Kumar B.S. Nemmani, Shri Vishnu College of Pharmacy from 2018. So has B.Pharm and M.Pharm degree from Andhra University Visakhapatnam. He has completed PhD degree from Naipur Mohali and subsequently postdoctoral fellowship from McGill University, Montreal, Canada. He has over 14 years of industrial experience in drug discovery and development and managerial expertise. Introducing our principal, sir, Dr. K. Prasad, completed his B.Pharm and M.Pharm from Andhra University, PhD from Andhra University, and worked as professor in HKE College, Gulbarga, and serving as principal from 2011. With this short note, I request our director, sir, to give a welcome note.
Greetings to everyone and welcome to day 5 of international webinar on drug discovery and development. At this note, I'd like to welcome you all and thank you for being part of this webinar series on drug discovery and development. I, on behalf of the SVS management, that is Chairman Sri K. Vishnu Raju Garu, Vice Chairman Sri Raichindan Rajgopal Garu, and our Principal Dr. K. Prasad Garu, and on behalf of all the staff of SVCP, welcome to this webinar series. I'd like to thank all the principals and the senior staff who are constantly supporting every event that is being conducted by SVCP. And at this juncture, I also like to thank Mr. Anil Kumar, the Organizing Secretary, and Dr. Sujit Kumar, the Program Coordinator, for nicely arranging all the requirements for this webinar series. I'm very happy, delighted, excited, and to introduce today's speaker and welcome for the webinar series, Dr. Gatta Srinivas. Can you keep the video on, Srinivas, so that others also can see you? So I'm very happy and proud for three reasons in welcoming our guest speaker. Srinivas, can you keep your video on, that photo? Yeah, I'm very happy and glad to invite the today's speaker for three reasons. The first reason is he is alumni of Andhra University, from where I also belong to, and our college is associated, Vishnu College of Pharmacy is associated with Andhra University, though we are an autonomous institute, but we get the degree certificate from Andhra University. That's the first reason I'm very happy to see the today's speaker. And the second reason is he is also alumni of NAAC, again, where I also belong to. And the third reason is more importantly, I'm very excited about is the research areas he has done in the past few years in the Western world. The research areas he worked is in antibacterials, and then he worked on asthma, and presently worked on immuno-oncology, and presently working on monoclonal antibodies for cancer. And the asthma and the cancer research, a lot is happening. The game has changed over the past 10 years or so. And he has done a great contribution in both the fields. And both are my favorite research areas. So I had many interactions with him during this period. I'm looking forward for more and more interactions in those areas as they are changing the lives of many people across the world. And prior to that, he did his PhD in electrophysiology, and as many of you, it's a very tough area, and he has done an excellent job. And I did contact him regarding some of the studies when I was in industry working on ion channels, and it helped me a lot. And the current role that he is doing is really, really interesting. So it's clinical research. So with this background, I requested Dr. Datta to talk on the clinical development. And we are studying in clinical development very easily, phase 1, phase 2, phase 3, and phase 4. We know very basics, and we don't know the intricacies and the nitty-gritties in doing those studies. So what is so unique about Srinivas Datta is he is a clinical scientist. He's not a typical clinical researcher who just design and follow the protocol. So he applies the science in designing those protocols and applying it and implementing it also. So that way he goes to different sites all over the world. And I can say without any hesitation, in India, Taj Mahal is the most photographed picture. And from Naipur, I think Srinivas Datta is the most photographed Naiparite with another Naiparite. So whenever he visits the sites in different parts of the world, he make it sure that he met another Naiparite. I think it's so unique about him. And he shares the same with all of us, and we are proud of him, the amount of energy and the enthusiasm and the way he keeps in touch with the alumni. And he has so much affection for all the places where he worked. I think this is a very unique thing with him, and I think we all should try to mimic not only science that he has gained, and also some of the unique characteristics. Because the networking is the most important. I asked him one day, how you are able to manage being in a senior position in the corporate world, and how you are able to meet this many people, how you are finding time. Then he told me, see, this world is moving to network. I learn a lot by interacting with a variety of people, and it's a quite good learning. So I make it sure that I find time to interact with the people. I think this is one message I learned from him. And I'm also now starting networking at this webinar series is one way of getting networking and connecting the campus with the different parts of the world. So I thank you, Srinivas, for really accepting our invitation. And I know you are very, very busy. And till yesterday also, he has not prepared the slides. So you can imagine how much busy he is. So he's, since he's committed, he made it, and he's addressing us, all of us today. So once again, thank you, Srinivas, for accepting our invitation. And there will be a formal introduction of you by the organizing secretary as well. This is my personal connections, which I want to deal with all the students. I want all the students and all the participants of this webinar, or their faculty or students. The networking is the mantra, and maybe we learn certain things from Srinivas today as well in this aspect. And also welcome at this note who has just joined now for this webinar series. And it is very stimulating for us to see the kind of interaction the students are doing with the guest speaker in the past few webinars. I expect the same thing to do so today as well. And because these are the people who are really doing on the job, and they can tell you which is not there in the textbooks, which is lying behind those words and sentences in the textbooks. So best way of learning is this. So try to use this opportunity, and same way also request all of you to give your honest feedback on the webinars, the way we are organizing it, and also let us know what you want to organize, and we will organize such more and more such webinars to make it more meaningful, useful, and interesting for your future careers. So with this note, I once again thank Dr. Srinivas for accepting our invitation and welcome you all for this webinar series. Over to Anil.
Introducing our speaker today, Dr. Srinivas Dr, Director and Clinical Scientist, Genmab, New Jersey, USA. Education, so he's completed his MS Pharmacology and Toxicology in NIPER, India, 2001. Masters in Clinical Research Organization and Management in Drexel University, Philadelphia. PhD Pharmacology at North Dakota State University, 2006. And his experience in various designations, like he's presently working as Director and Clinical Scientist in Genmab. At GSK, he has various designations. He worked in various designations like Clinical Scientific Leader, Immunology Oncology. So Associate Director, Clinical Development Manager, Scientist in Clinical Development, Clinical Pharmacology Operations with Biopharm and Infectious Diseases, Principal Scientist and Investigator. So with this wide area of expertise, we are sure that this session will enrich participants' understanding over the topic. So today's topic being Introduction of Clinical Research in Drug Development, special attention on Tisotumab, vedotin's GEN 701 study. So with this short note, I request our today's speaker to start his technical session. Thank you, sir.
Thank you, everyone. So first of all, thank you everyone at Vishnu College of Pharmacy and Dr. Kumar for inviting me. So the core mantra for me is like sharing is caring. Like as a kid, we all talk about like, sharing is caring, and then we have only one son. So I tell all the time, you share the things, whatever you have with your friends and everyone. So I feel like, sharing for a scientist, if you need to share what you know. That's how you grow the science. So I feel like when Dr. Kumar asked me to present something, so I thought like immediately, yes, this is the time that I can present something to everyone. So thank you very much for this opportunity. And I am really looking forward to learn from you too. So I'm going to share my screen right now. So I thought to present my study, what I am doing, what I have done. But later I realized that it would be great if I can give a little bit more details about clinical research before going to the actual clinical study that what I'm doing. So in that way, you all will understand where I'm coming from, and then at the same time we all can apply. You can see where we are applying that thing. So my talk so to start my presentation, I thought it should be fun, right? So what is clinical research? Some people think that just simple thing. So like this cartoon says, but it's not that simple. I wish that this is that simple. Hence, I'm going to talk a little bit more in the introduction of clinical research, ethical issues which were involved in clinical research, which are involved in clinical research, different phases of clinical trials, and what are those lessons learned in that different phases, phase 1, phase 2, phase 3, and clinical trial protocol contents and clinical trial designs. I'm not going to go deep in every aspect because the whole contents that I'm mentioning here, it will be a year-long course. So I'll go touch and then leave. So if you have any specific questions at the end, we can discuss. And the other important thing is, which I'm going to talk, is clinical trial staff at the investigator sites and then sponsors are CROs. Because majority of you are some of you are willing to go to clinical research. But when I was trying to move into clinical side, I would like to move to clinical side. And however, people ask me, what do you want to do in clinical? Clinical research has several aspects, several roles, but what do you want to do? So I would like to give that information in the first part. Coming to the second part, after interval, we're going to talk about my study briefly, which I am currently doing at GenMab. And I'm going to mention who are GenMab and what we do in the part 2. So let's buckle up, and we're going to go and have a nice ride in clinical research. So these are the 2 papers that we recently published. Yesterday, I did attend Dr. Sambareddy's talk, and then I really liked the way that he did show his papers. And then hence I'm starting my presentation with my 2 papers we recently published, one in Lancet Oncology and other is Clinical Cancer Research. This data I'm going to show at the end, that is in part 2. One thing I would like to show, let people know that majority of the preclinical research, when we do preclinical research, when we publish, either we will be in the first author or second author or the last author. So it's kind of, whereas in clinical, it's very difficult to get the first author paper because most of the times the investigators are the ones who is running the study. And then people from sponsor or CRO, they are overseeing the study. They design the study. They let the study. They give this study to the investigator, the hospitals who are going to run this study. So before going to the presentation, I would like to mention one more thing. When I was a bench scientist, like Dr. Kumar has mentioned, I was in the lab for almost 4 years at the bench, trying to discover various pathways in respiratory and neuronal pharmacology, kind of like somewhat unhappy because whatever I was doing is in the preclinical side, and whenever I go to a patient-focused seminar, they talk about this drug acted in this way, these many patients got benefited. I was like, oh, I'm in the lab, I'm not able to help, I'm not doing directly anything to the patient. So I really would like to move to clinical side. It was very hard path. It's people will ask the chicken or egg question, like, do you have any experience? Yeah, no, I don't have experience, but if you give me experience, I'll take it. But they will not. I mean, it's very difficult for them to give a clinical study to an inexperienced fellow. So it's a difficult thing, but it can be doable. It's all you being in the driver's seat. You can you know where you want to go. You can push hard. You aim, you dream, you aim, you design, you success. So 2 drugs that I worked during my clinical, one is dolutegravir on the left side of on this slide. It's an integrase inhibitor, which was approved from GSK. I worked on this molecule, hence I put the picture here. Of course, on the right side is the respiratory molecule in eosinophilic asthma. I worked again on this molecule. It's called mepolizumab. It works on IL-5. These 2 are the kind of, I shouldn't say I'm the only one who worked on this, because to get a drug, to market a drug, it takes a village. No, it takes a city. Sorry, it takes the whole world. So what I'm trying to say is, like, I'm glad, I'm happy that I was part of in the development programs of these things. So what I did is, like, I did work in phase 1, from phase 1 to phase 4, including medical affairs studies, and I gathered really good experience by working on different things. Sometimes from preclinical, if you go to clinical side, you feel like hey, this is just a 10th standard kid can do. Why am I doing this? But it's a process from preclinical to clinical. You're going to miss the lab, but it's an awesome journey. So let's move on to the part one. So this is the introduction where I would like to show what is clinical research? Clinical research composed of clinical trials, mainly whatever we do clinical trials in human subjects, that's a clinical research. It's basically from preclinical you do in the animal species and the cell line, whereas the clinical research is composed of the clinical trials in the, human subjects. The important thing with the clinical trials is like one needs to have the hypothesis. If the hypothesis is strong, you basically select the endpoints. If you don't select the proper endpoints, you may not be able to achieve, or you may not be able to test your hypothesis. So having those two in your mind, end goal, is a crucial thing. Think about those hypotheses, what you really want to test, because if we are involving human subjects, other lives, so one needs to be very careful with the safety, and especially safety. Efficacy comes later. So safety and quality are the crucial aspects of clinical trials. So prior establishment, so before coming to the clinical trials, we all know that like we're back in our hometowns or places, people say, oh, take this leaf or take this stem, eat it, it will help. But those are like, before clinical trials. You cannot extend things based on experience, observations. You cannot consider that drug or that test article is working. You still need to do clinical trial. So what we used to do is different. What we need to do is different. Currently, clinical trial is the gold standard to make it market, to make a drug or test article to market. If without clinical trial, you cannot do anything. It used to be possible with some Ayurvedic compounds, but not anymore. What are the limitations? It's not that like clinical trial solves everything and anything. It has its own limitations. So if the sample size of your patient or subjects you're taking not optimum, you're basically shooting yourself. The statistics, the power, it needs to be maintained with the number of subjects that you are testing. If you're not doing a correct balance between your power and then your sample size, you're basically not testing the proper way. So for that, the sample size may go, may not be enough. And on the other side, you're basically telling the drug is active when the drug is not active. So hence, one needs to design a proper clinical trial with the end goal in the mind, and then understanding that we are testing this drug in human beings. Like I mentioned, well-designed studies yield interpretable results. If you don't design properly, it's basically you get result, but you can't be able to interpret anything. So some questions, I put some questions here, and these are like not the only questions. There are some. So I feel like these are a very important question. So one needs to reduce bias. Sometimes people come and then they come, this will work, that's it. Without any scientific rationale or without any prior data, they'll say this will work. So you need to come and then you need to mention all the things. You need to provide the background why this won't work. So bias is a huge thing, irrespective where you go, industry, academics, wherever you go, personal bias is the biggest issue. Second thing, one needs to, what are the objectives that one needs to be answered? If you don't, hypothesis, then followed by the objectives, and then what procedures that you are going to follow to ensure the patient safety, which is a crucial thing, which I'm going to mention in upcoming slides. Why patient safety, and then why we need to protect people, participants, study participants. And then the other thing is this trial feasible or practicable? Based on, you cannot say that I need to test 100,000 or 1 lakh patients to get this data. It's not feasible, it's not practical. So one needs to understand that the balance, strike the balance between the practicality and then feasibility. And then the target population. So you need to have those target population to test. If there is no target population, you even though you have great drug, it's no use. And then you cannot extrapolate or interpolate your adult drug. The drugs which are approved for adults cannot be extrapolated to the kids, because kids are not small adults. So you need to have the target population for your drug. If the drug is for geriatrics, you cannot use that drug for pediatrics. So you need to have the target population. And what kind of trial design? Not every trial design is valid for every study. So one needs to know before even design the study, what kind of trial design that you want to choose. So I'm going to mention the trial design. We do have the part in part one. I have some trial designs which I'm going to mention. Is this trial ethically correct? Very important. So we all, irrespective of preclinical or clinical, ethics is crucial. One needs to follow, ethics should be bedrock. If that base is strong and foundation is strong, we can create both preclinical and clinical experiments strong. So irrespective of preclinical or clinical, ethics is very important. And then trial participant, do we have enough trial participants? I'll give an example. And then you have an ultra rare disease, but you want 1,000 patients to run your study according to your power. It's not possible because there are no 1,000 patients. You may have only 100 patients throughout the world. So one needs to understand that one too. What do we know about the drug currently? Safety profile. Based on the preclinical, based on the translational biology, translational medicine experiments, even though you don't have clinical data, but you can use the preclinical data, translation data, and then kind of figure it out, like what kind of safety profile this drug will have. Not every time you can be successful, but sometimes. And what's the correct starting dose, which is a $1 million question for every new molecule. This exercise all of us will do at industry. What is the starting dose? We do have experts, PK experts, pharmacometricians who uses the different modeling and other aspects to design starting dose. So let's go back to the history, and then you probably might have studied all these things. Some may not. So I thought, I would like to hold your hand and then take each one of us through this slide. So please bear with me if you haven't, if you knew all these things, previously. The one thing that history, let's go back to history, how this clinical research evolved. Nobody has from the start decided, okay, we'll do clinical research based on XYZ guidelines. It's all evolved. Even FDA evolved. These rules and regulations evolved. We evolved. So this research is purely empirical. So 1937, in US, there was a huge issue happened. It's for sulfanilamide elixir. At the time, sulfanilamide was basically useful for antibacterial. So they wanted to try different formulation because they needed a good diluent. However, they were not able to find good diluent. So this company called Messenger Company in Tennessee. The company's owner was Messenger, and then he hired a pharmacist / chemist, and then this chemist decided to use diethylene glycol, and then which gave nice taste and everything. It's soluble, and then it looks good. So basically, they produced sulfanilamide with diethylene glycol, and guess what? So because diethylene glycol kills your renal cells. So renal failure happened. There were 107 people died, and majority of them are kids. So all of a sudden, FDA was there. It's not that FDA was not there. FDA was there, but FDA was not testing the safety of drugs. So FDA all of a sudden realized, okay, the new drugs needs to be tested for safety too. Hence, in 1938, the Food and Drug and Cosmetics Act passed in US. Basically, it became strengthened. Fast forward, 1940s. So during the Second World War, Nazis, Hitler has a special wing for conducting experiments. It's very difficult to even read what they have done. So it's a big disgrace on humanity. What kind of experiments they did in the 1940s? What they did, they considered themselves our superior Germans, Nazis, and then every other race they considered are like inferior, especially Jews. So whenever I'm going to give a couple of experiments. The big chief for their notorious guy is Joseph. His name is Joseph. Last name is I forgot. Mengele. Sorry, Joseph Mengele is the doctor who had the angel of death. They call him angel of death. So he used to nab twins. Wherever there is a Jew or other community has twins, they used to nab those twins and then test on one twin kid or one twin adult, and the other twin is a control. They used to amputate, they used to give injections where the subjects didn't even know what they are, and they used to kill. It's kind of nothing but murder, genocide. And then they used to check the difference between both twins, like one with the injection of different things and the other one is without. So I'm talking about 3,000 twins. So, and the total number of deaths based on these Nazi experiments, humongous. It's several hundred thousands. So what happened with this? There's a Nuremberg. The Nazis started losing the war. Hitler was dead, and then Mengele, some reason, escaped to South America and then never got any fair trail. However, the good thing happened is the Nuremberg Code in 1946 came out, and then what they did is like from now on, Nuremberg Code says, subject participation in clinical trial is voluntary. If you don't want, you are not supposed to, nobody's going to coerce you to participate in the study. And then researchers must be qualified, whereas in the Nazi army, they were not qualified at all. The study design must be justifiable and scientifically sound. So I recommend this book. I found this book reference for you guys. So if you can find this book, definitely read, and then you will realize how bad and how dangerous experiments, how horrific experiments that they have done on human beings during Second World War. So we several of us are familiar with thalidomide incidents, and then we probably might have studied in school books, college books, university books. In 1950s, 60s, post World War II, what happened is like everybody's happy or everybody's not happy. People wanted to take a lot of sedatives. So at that time, this company produced this thalidomide, and they advertised this is a great sedative, which is non-barbiturate sedative. And they even were prescribing for pregnant mothers to prevent morning sickness. There are 40 plus countries approved this drug. However, I would like to bring this wonderful lady here on this slide. Her name is Dr. Frances Kelsey. She was a reviewer at FDA. And then basically she was reviewing this thalidomide application for marketing. And then she was not comfortable in approving. And then she said, no, I'm not going to approve, even though she was getting kind of pressure from her superiors and then from the pharmaceutical company. She did not give up. And then basically she didn't approve. guess what? She basically saved several babies and several people in US. Hence, she got this the highest honor in US from President Kennedy. And then one more thing I would like to mention here about this wonderful woman, Dr. Kelsey. She's a Canadian American. She came from, she was born in Canada. She did her bachelor's, master's from McGill University, where our Dr. Kumar also did his postdoc. So I thought maybe, great people come from McGill. So other thing, what happened after post the thalidomide incident, the Keffer-Harris drug amendment happened in 1960. So which basically further strengthens any manufacturer, they need to show their product safety and efficacy before marketing. So again, for the strengthening of safety and efficacy aspect in drug approval. So my last example in where we did do wrong in clinical research, this is Tuskegee experiment, which is a big bad mishap, bad incident in my opinion that US government did. So they have designed a study to understand syphilis natural course. Before HIV, syphilis was a huge issue in US or in the whole world. There was no medication. So these investigators wanted to know how the syphilis natural course occurs in humans. The study was correct. It's a good study. Natural course is understanding natural course of a disease is a good thing. However, they have done several mistakes. First one is they have only recruited African Americans. There were no white Americans. Second thing, during this, the whole study was designed for 6 months. However, the study went for 40 years, 40 years, from 1930 to 1972. During this process, penicillin got approved. Penicillin was approved for syphilis. However, none of the study participants were allowed to take that penicillin, which is barbaric. They were getting their burial allowance. They were getting their health allowances. Never. However, they did not get anything, any penicillin for their disease. So what came out of this? So I recommend again this book. If you have time, please go, please read this book to get to know a little bit more Tuskegee experiment. What came out of the Tuskegee? There's something called IRB or ECs. IRB means institutional review boards. EC means ethics committees. Most of the times, ethics committees will be in Europe, and then IRBs are in US. Every site, every institution, whoever is running clinical, IRBs needs to say yes. They need to check whether the safety versus efficacy is a valid thing for those participants. So irrespective regulators, irrespective of the sponsors, even if we say, yes, let's do study, if IRB says, no, it's not valid, we cannot be able to do anything. So this is one more check where they will check, they will inspect the whole protocol. They will see the positives, negatives, what are the advantages that these patients, these subjects will get while in participating in the study. So what I'm trying to say is like, it's not like a day or, in a year or 10 years or 20 years. You can see we evolved, the regulations evolved. Every day there is new thing. So clinical research, that's one thing. That preclinical, when you're in preclinical, you read about papers, what's happening, in different labs. In clinical, in addition to that, one needs to be, needs to keep updated with the regulation. What's happening in different countries? What's happening with the different regulatory authorities? So that's one thing that I recommend all of you. If you are interested to do clinical research, please. Sorry. Okay, so we got to know that there are several things that put in place, and so there may be any fraud. Am I right? No. Coming fast forward, 1990s, we have all these wonderful things. Okay, let me go back. I missed one important point. So there's one more thing after the Tuskegee happened. That is all these countries, all these regulatory authorities are trying to work more and to create a regulation. There is a harmonization procedure. Harmonization happened for the clinical trial guidelines. That is called International Conference on Harmonization, ICH. You probably, if you want to be in clinical, you probably will hear ICH name probably every hour once or every 10 minutes once. ICH GCP, GCP means good clinical practice. These are harmonized regulations. Most of the times, FDA has their own, WHO has their own, EMA has their own. But most of the times, these ICH ones are kind of a Bible, Bhagavad Gita, or Quran for reference. So what's happening in the recent past? We all think that we have so many regulations, so many things, so there may be there won't be any fraud. No, like I mentioned, ethics play a vital role. If you pledge with yourself, your conscience, that I will not do any fraud, I will not do any unethical things, both in preclinical or clinical, or anywhere where I go, that will help all of the researchers, in my opinion. Sometimes you see really great investigators, researchers, but still they fail at that basic point. So majority of you probably are the early stages of career, or mid stage, are like about to start your career. So definitely pledge yourself, like what is the thing that you really want. Bedrock, my fundamentals, my base is ethics. I don't want to go into any fraudulent activity. So I'm going to give two examples here in this slide. There are two people came into my attention that I would like to share with you. That's a Professor Bezwoda from South Africa. He is a great oncology researcher. So he was publishing several papers, and then till that time, till 1990s, this allogenic transplantation of your own cells, or giving your own cells to you, or for hematologic blood cancers, was helpful. What he did is, like, he published some papers saying that it's same way or much better, helpful in breast cancer. NCI, National Cancer Institute, tried several times the same experiments. They were complete failures. So they were not able to come close to whatever he reported. And then he was happily giving presentations in several meetings. And whereas they were not able to do any replicate, doing the same, getting the same data anywhere. So what they did is they investigated him, and that they found whatever he did was not proper. There were no subjects in some cohorts. He basically took the data, and then they found him, and then basically his whole research life is gone. On the other one, I would like to give an example. Some of you may end up going into clinical operations. And then most of the times in clinical operations, you think that which site is going to open fast, which investigator is active. So let's work with them and then get more subjects in your clinical study. This investigator, Robert Frides, is from US. So is a favorite investigator for several people in 1990s because he was recruiting several subjects in several studies pretty fast. And then later they found in the investigation, there were no subjects. He was just putting the dummy subjects. And then whenever there is a need, what they found is like he hired a proteinuric patient and then collecting his urine and then creating dummy subjects, saying that this urine come from several other subjects. So what happened ultimately, this fraud tarnished the science, institution reputation, and their reputation. And then the important thing is we are talking about a valuable life, human life. They think that these subjects, these patients, they don't have any other option. They come to the study thinking that something will be helpful. Some more days of life will be granted by participating in this, some kind of hope. They have basically destroyed that hope. So it's not only destroy your career, it destroys other life too. So I recommend everyone to take a step back when you are doing your clinical or preclinical studies. Think about all these prior experiences, ethical lessons, that what we learned and what we saw in other investigators too. So let's move to what types of phases in clinical trials. So we got the ethics, we got the where the fraud happened. Now it's like different phases of clinical trials. The phase one, you probably might have read this in several textbooks. However, I'll go in a different direction. Phase one, mostly in healthy volunteers. However, it's not always. In rare diseases and oncology, in even phase one will be, we recruit patients. And then most of the times 20 to 100 subjects we recruit. And the main thing from phase one is the safety, whether the drug is safe. And then we'll find what is the maximum tolerated dose. Sometimes you may not get that maximum tolerated dose, but that's the ultimate aim, to get the how much dose that you can give to human beings, to healthy subjects, or sometimes patients. So phase one studies will provide several informations. I would say, if you are interested in preclinical, and then if you are interested in translational biology or translational medicine, phase one is an exciting area. You can use your science, you can apply the science, whatever you read in your textbooks, and then see, you can design different biomarker analysis, or different efficacy analysis, or safety analysis. So it's an exciting area. I mean, if you ask me what's my favorite, this is my favorite. Phase one and phase two are my favorites. What do we get out of it? We get the safety, we get the PK, pharmacokinetics from this drug. How do we design? And then an important area that if you want to go is pharmacometrics, pharmacokinetics, where these are super smart mathematicians who take all the biological data, and then they will put into the equations, and then they come up with the starting dose in human beings. So very, very exciting part, PK/PD correlations, pharmacokinetics, pharmacodynamics correlation. If you are interested in that area, I highly recommend to pursue in that area. There are several jobs. In every company wants to design clinical trials, and they want to find out what's the starting dose of their drug. So that's a very hot area right now. And how can we design, how can we get the starting dose? There are two ways that they generally do it. One is Noel, other one is Mabel. Mabel incorporates the in vitro, in vivo data more. Hence, FDA and then other regulatory authorities these days are more interested for biologicals, Mabel, than Noel. I'll give more examples where Noel failed in my upcoming slides. So what are the key considerations for the sponsors that, what are the things that you can get? One is the selection of starting dose, and then you will get the adverse events of the special interest. And then primary correlation. If there is a biomarker, I'm going to give a hypothetical example. If this drug is causing liver enzyme elevation in animals, we can test that. Okay, is it still is same thing happening with human beings? So you can test that in phase 1. And then other thing, one needs to be careful with the phase 1. Not everyone is a phase 1 expert. Like me, some investigators. I'm not an MD or a doctor. I'm a PhD doctor, not the doctor, MD. So we need to find people, MDs, who are really interested to do clinical research in phase 1 units. That is the crucial part, because not every investigator is specialized in that area. Other thing that we do in the phase 1, for most of the time, this is oral molecules or not large molecules. This is small molecules. That is SAD, single ascending dose, and multiple ascending dose. To make it easily understandable, I put this slide. You can see on the left side is the single ascending dose. Open triangles is the administration of dose. A, B, C, there are the 3 different subjects. So each subject will get 1 dose in single ascending dose, and probably A will get 1 milligram. I'm just giving a hypothetical to make you understand. B will get 2 milligrams or 3 milligrams, and C will get 10 milligrams. That's a single. You are basically escalating the dose. And the other one is multiple ascending dose. With the D, F are the different subjects, and then for the D, you're giving 1, 3, 10. You're basically escalating intrapatient. That's multiple ascending dose in the same subject. So this is one thing we test in phase 1 for small molecules. Coming to the lessons learned, this is a very important part. I'm not sure how many people know this. However, I recommend everyone who are interested to come to clinical research to learn from these 2 examples. There's the first example, it's TGN1412. In 2006, a company called T-Genero came up with this molecule. It was targeting CD28, and they wanted to test this for rheumatoid arthritis and then B-cell tumors. So they thought, in the preclinical experiments, they did not see any safety issues. So they took a dose of 500 times lower than their monkey tested dose, that is 500 times lower, and then they gave that drug to 6 healthy volunteers in the UK. The CRO that was running this study was Parexel. The sponsor was T-Genero. However, a huge thing happened, big debacle. The six subjects immediately had cytokine release syndrome, cytokine storm. People were shouting with pain. What they did is, like, they have given all the 6 subjects at the same time this drug. Even though this drug was like 500 times lower than what they tested in monkeys, still produced a huge cytokine storm. And then even they call this as elephant-like syndrome. So some people became like elephants. They puffed up, they became huge, and then some people, they lost their nails, their fingers, they amputated. The doctors had to amputate their fingers because of this cytokine storm, which I'm going to show a picture. It's kind of a simulated picture. It's not the real subject. However, UK's highest medicines and healthcare product regulatory agency, like FDA, they went back and they checked whether they have done anything mistake, that T-Genero has done any mistake while designing this clinical trial. However, they did not find any issue because they followed everything as per the book. They used the NOEL, they used 500 times lower dose. So for the design wise, they didn't find any issue. However, the CRO has done several mistakes. The trial conduct where this trial was happening, like I mentioned, generally phase 1 happens in one hospital or one specific unit. That place has so many deficiencies. The investigator was not specialist. He didn't know any, he was not a specialist in immunology. And there were, and the dose they selected was based on novel. And then they haven't done several preclinical in vitro experiments, not preclinical, some in vitro experiments such as co-cultures and other things they haven't done. So overall, this is a big lesson. The other lesson is like what people got out of it. You cannot inject the novel drug or novel asset or test article to subjects at the same time. You need to give some time from one to another based on the half-life. So this is the pictures. I mean, it's probably a little scary to look, but you can see on the left side, this is an Indian, actually he's an Indian, went to the UK to study, and then he participated in this T. Genero study. And you can see how he used to look, and then how he became immediately after the administration of TGN 1412. And then on your right, there's another study volunteer. You can see basically this subject lost, because of the amputation, he lost some of his fingers. The other example, fast forward 2016 now, a Portuguese company in France wanted to run the study on fatty acid amide hydrolase inhibitor. I think, again, I'm going to refer Dr. Kumar here. He has done extensive research on this area. So this company, Bial Portela, is a Portuguese company. They're not first in class. Johnson Johnson, other companies were doing finished their phase 1, and they were doing phase 2. However, Bial Portela started their phase 1. They finished their single ascending dose study because it's an oral molecule, no issues, no safety concerns, and then they started multiple ascending dose. When they did the 5th MAD multiple ascending dose cohort, they again gave the dose not at the same time, but around the same time. And then 4 out of 5 had severe, immediately hospitalized because of the brain injury. One subject passed away because of hemorrhagic shock, and then it's like a brain. It's like brain injury, the whole brain injury. The subject passed away. Why it happened? So again, people went back and then checked, why it happened. They used Noel strategy to start for the starting dose. However, in this, that's not the issue. The biggest issue, the non-specific antagonism or activation of other enzymes of this molecule in addition to the fatty acid amide hydrolase. So they haven't tested other enzymes, how they are getting affected with the high concentration. So when they give multiple ascending dose, they basically gave large doses and basically accumulated, and then that caused activating or inhibiting other targets other than FAAH, caused this thing. We had several issues post 2016. We were not able to recruit any patients in France. It's purely because it happened in France, and then they said they put all the regulatory guidelines around it. Of course, these are the lessons learned, and then we take those lessons and then incorporate those lessons. Regulators take these lessons, they'll incorporate in their regulatory guidelines. We will move forward based on these guidelines. Let's move to the phase 2 studies. So phase 1 studies, the chances of your molecule going from phase 1 to phase 2 are 70%. That means if you have 20 molecules, you have 14 molecules will go, or 7 out of 10 will go to the next level. In phase 2, its main purpose of phase 2 is safety again and efficacy, because we're going to test that in patients. Phase 1, majority of the times healthy volunteers. Phase 2 in patients. There again, phase 2 is divided into phase 2A and phase 2B. Phase 2A is mainly you still can test the 2 different doses, because in phase 1 you may not be able to find the exact dose. You may find like between top dose and then 1 level below. You probably not sure which one will be active. So you can test in phase 2A those 2 doses, and phase 2B is basically you will almost finalize your dosage, or dosing pattern, or your dosage, everything. At the end of the phase 2, if it's in US, you generally meet with the FDA. It's called end of phase 2 meeting. And then basically you take the data and then you show the data to FDA. This is what we got, and this is what we wanted to design for phase 3. Do you agree? And then basically most of the times, because it's an expensive process, phase 3, it's a time consuming. So FDA will give the guidance. Okay, this is what we are thinking. I mean, sometimes they give direct guidance, sometimes they don't. They give hints. So it's very difficult. The regulatory team are experts in this area to find what they really want, what they in between the lines, what they're trying to say, how we can avoid questions from the FDA or EMEA later. So they play a key role. Yesterday I heard a question on regulatory. It was a very good question. So what's the general percentage that phase 2 to phase 3 molecules go? Around 33%. So if it's 7 out of 10 comes from phase 1 to phase 2, now we are talking about 33% will go. So that means probably 2 molecules from phase 2 to phase 3. Once we go to phase 3, I'm going to talk about their attrition. So before going to phase 3, there is one lesson from phase 2 I would like to share with you. This is a company called Hanmi in South Korea. What they were doing is like oncology research. They found a molecule called olmetinib. It's a 3rd generation EGFR mutated for lung cancer, mutated therapy, mutation positive lung cancer therapy. They got approved in South Korea. They submitted that data with their partner in US, and then FDA was thrilled to see the data, and then FDA immediately gave their breakthrough therapy designation. That means basically they gave a special status. Yesterday, Dr. Samaradi has also mentioned different types of special applications. So this is one special application, BTD. So they gave that. However, South Korean regulators went back and checked about the safety incidents that were occurring with the olmetinib, and they found out Hanmi, the company, did not report their safety issues, especially there's something called Stevens-Johnson syndrome, not on time, even though that was a severe adverse event. There's a timeline for severe adverse event that we as a sponsor need to report to the regulatory authorities. If it's SAE, we need to do that in 24 hours as soon as we know. So they did not do that, and they found there was 1 death, and that was reported very late. So ultimately, whole thing gutted, the whole program gutted. The olmetinib in South Korea stopped. They're not doing anything, and then in US, if I'm not wrong, they're not. Basically, they stopped the development. So very important aspect, again, coming back to our own starting points like ethics and then fundamentals. Other thing is transparency. You do not think that like nobody is looking, or in my opinion, like these inspectors, these regulators, they exactly know what can happen, what will happen with your drug, because as a sponsor, we know that about our molecule. It's like our baby, so we know exactly what's happening. But as a regulator, they receive these kind of applications from whole world. So if if FAAH, other people, the other companies' data, they can go back and see, okay, this FAAH molecule is causing this thing. Why didn't we see this thing here? So then they'll ask questions. So it's transparency is the key, and then as soon as the safety reporting is also key. As soon as anything happens, we need to let any SAE, we need to let regulators know. So coming to the phase 3 studies, these are global trials, and most often, very important studies. Patients range from 300 to 20,000. However, some ultra-rare diseases, you can get approval even with the 10 patients. That's because the availability of those patients, occurrence of that disease is very low. For example, spinal muscular atrophy and other diseases. So what are the lessons that generally you learn from here is it's an expensive thing to do phase 3. Most of the times phase 3 fail because of the high placebo, or if you don't select your patient population properly, you're basically getting the placebo effect or no effect in your patient population. Expensive. So if you run a phase 3 and other phases, overall the cost will be $1 billion per molecule. It's an expensive thing. How many molecules can go from here to the marketing approval? So based on phase 3 data, generally FDA gives approval. They may ask for the confirmatory study, like they ask 2 phase 2, but sometimes they give approvals even with 1 phase 3 in oncology. Generally 25% of molecules will move from here to the marketing. That means we talked about phase 2 to phase 3 were 2 molecules, and then now we're talking about 25% of 2. So we're talking about less than 1. So if a company starts unusual process, 20 molecules to start with, we may get 1 molecule in marketing by spending $1 billion. So innovation is not simple. Innovation is time-taking. Innovation needs teams. It's not one person. It's highly dependent on every person. Every person is important in clinical drug discovery and development. No one is small, no one is big. So I'm going to skip the lessons learned. So the other types of studies that I'm going to mention is investigator-initiated studies. Most of the times, these are run by investigators. Investigators at a hospital, they would like to try, they would like to test a molecule in their patient population to see whether that's working or not. Instead of giving off-label, they can follow this path. They can get the IND, investigate a new drug application with FDA, and then they can conduct the study. For the drug, most of the times, the drug will be given by supported by pharmaceutical company. These are sponsored by investigator, supported by pharmaceutical companies. What will we get out of these? We'll get additional data. For example, if our company does test that drug in X indication, this investigator may try in Y indication, and then if they find some good data, if they get good data, then we will take that and then run a big study, maybe a phase 3 study or phase 2 study on that. Another important aspect, the clinical trial protocol. What do we have? Clinical trial protocol is a compendium of everything what one needs to do. It needs to be developed before any application. Most of the times, if you want to run a study in US, you need to submit an application called IND, investigate a new drug application. For that, you need a protocol. Excuse me. If it's Europe, you have to submit a clinical trial application. If you want to read a little bit more about like what a protocol has, what are the contents, what are the subsections, I highly recommend this website called Translate by Pharma. They have common protocol template. You can go and then you can read what are the contents of the protocol. let's go back to the let's move to the clinical trial designs, which is a crucial aspect. It's a big course. I would say the whole year we can talk about clinical trial design. However, I'm going to mention a couple of important clinical trial design. The first one is parallel design. In parallel design, you basically assign one group, the new drug or a drug to one group, and the other one is like placebo. So you basically compare these two. These are not that rigorous than randomized control studies. So when I said randomized, you basically assigning the treatment to subjects in a random manner. It can be a single blind or it can be double blind. Single blind means a subject or patient, they don't know what they are getting. Double blind means both the investigator and sponsor and patient or subject, they don't know what they are getting. The code will be broken at the end of the study, or if there are any safety issues are happening. So what I'm trying to say is the parallel designs, they are not as strong as a randomized control study. If somebody asks what is the gold standard clinical study design, it's a randomized control study. So that's why if even if we see most of the times if we see any clinical data right now, everyone is talking about COVID. We question about the data. Patanjali has mentioned that they have a complete cure. So as a clinical scientist, I first ask, what's their data? How many patients? Whether they ran a randomized control study. If they haven't run randomized control study, I wouldn't believe till I see the data. So other designs is within patient design. The first one is here we're talking about crossover study design. You can see in the picture there, part A. Treatment A, there are two periods and two treatments to make it easy for you. So the treatment A in the period 1 will be switched to treatment B in period 2. Vice versa, in period 1, treatment B will switch to treatment A. Similarly, the Latin square design, if it's more than 2 periods, and then more than 2 groups, you basically use the Latin design. That's here in the picture, you can see B there. And the other thing is like C, which I'm going to explain in factorial designs. If you have more than 2 drugs, then you can use factorial design. These are mostly used in mortality studies. You can see the C here in this picture, where you can group 1 has cisplatin 2 drugs, group 2 has 3 drugs, group 3 has 2, group 4 has 3. So how can you compare all of them together, basically using a factorial design? The other thing these days, everyone talks in oncology, and which is a very useful, powerful thing, is precision medicine. Based on biomarkers, you can design studies. There are 2 studies right now is very famous. It started 4 or 5 years ago. There's something called umbrella trial, where they select one cancer, for example, here in this example, lung cancer, but different mutations. So they have the PD-L1, EGFR, and other mutations, and they give different drugs in the same study. The other one is called basket trials, where you have different tumors, but you use one drug. So what are the advantages with this? You are basically expediting the clinical research by combining different things at the same time. It will save a lot of time. It will save many lives. So these are the current trends in oncology research. So this slide is again, I can talk for the whole day, but I'm going to go touch and then go. key staff in clinical research. At the investigative sites, hospital, it can be hospital or clinic or an institution. The main person will be there is a principal investigator who is responsible for the ICH-GCP at that site. And then principal investigator make sure the drug is, everything is working, everything medically is correct. And then trial is conducting according to ICH-GCP guidelines. And then the other one, the second person is clinical research coordinator. Some places we call them clinical research managers. They are the ones who operationally check everything with the sponsor and CRO, that they are, their patients are lined up, that the patients are getting every, kits, and the patients are signing the correct consent forms, because informed consent form is a crucial document that every patient has to sign before entering. Every patient or every subject, they have to sign before entering into the study. The third role that I'm going to talk about is a data manager at the site who enters the data. If there are any issues with the data entry, or there are any discrepancies, they are the ones who takes care of it. At the sponsor side, on the CRO side, I am currently working in the sponsor side. I'm a clinical scientist. I haven't mentioned two roles here. Please accept my apologies. One is medical monitor, medical lead, and the second one is safety physician as a clinical scientist. Those two are not here. So what medical monitor does, again as per ICH-GCP, we, a clinical scientist and medical monitor, work very closely, and we make sure the subjects are getting, we design the protocols, we make sure the protocol is executing properly according to ICH-GCP guidelines. Are there any issues with the data? Once they are collecting the data, or once they are seeing the patient, there may be a safety signal, there may be something else they are seeing. So they come to us, and then we basically discuss very closely with these investigators, we weekly, daily sometimes. And we other roles such as safety physician, who specifically look into safety aspects. A safety scientist is the one who tries to find trends. Data programmer, stats programmer, they program the whole data pathways. And pharmacometricians or pharmacokineticists is the one who design the early the dosing and PK/PD collections, and if there is any anti-drug antibody generations are happening with the drugs, they're gonna work on that. Medical writer are the ones who help us writing the protocol, informed consent forms, and any other documents, ICF or any other document, they will help us. And CRA is the heart and soul of sponsor and CRO side. They are the ones, CRA is a clinical research associate is the full form. They are the ones, they go to the investigative sites, they work with the investigative site. They're kind of a liaison between sponsors or CRO and to the investigative sites. So if your CRA is good, if that CRA knows the tricks and trends of business of clinical research, you are in safe hands. So having good CRA is a wonderful thing in clinical research. And CMC lead is the person who makes sure the drug formulations are, drugs are reaching into the depot, or the formulations are making, any issues, any particles, or any batch issues. They are the ones who make sure the operations and then formulations. Project manager makes sure everything is happening at the timelines, no issues with the delivery. We are not spending too much money. We're not delaying. They're gonna find that. They're gonna take care of that. And translational medicine lead who makes sure the biomarkers aspect of it. So a small break at the end of the part one. I think I'm running really late, so I'll be quick on the part two, just to make you chuckle or just to make you wake up. I put a small cartoon here. So let's go. Let me go to the my study update. This is the study that I am doing for the last 4 years. Again, clinical research takes time. If you're in clinical research, you probably might not have seen all the you might not have seen all the aspects of clinical research. I'm in clinical research for the last several years, and I haven't seen everything in clinical research. I'm still a student. I'm still learning several aspects of it. So before going to the study date, I would like to give a little bit information about our company. Our company is founded in 1999. It's a Danish company. Our head office is in Copenhagen. This is Europe's biggest biotech now. Right now, 550 employees. We have employees in 4 countries: Japan, Copenhagen, Utrecht in Amsterdam, Utrecht in the Netherlands, and Princeton, where I am working in US. We are a very successful large molecule company, and we do have several molecules both approved with the different companies, and then some of them are in development. Right now, we just signed a deal with AbbVie, Abbott, 50-50 partnership with 4 billion molecule pact. So what is Tisotumab vedotin? It's an antibody drug conjugate. What preclinical experiment? Everything starts with preclinical. So even in clinical, if you understand what preclinical experiments are really, why they're designed, what is the purpose, then you can be very successful in clinical. So the fundamentals are definitely from preclinical. If you work in the lab, I think you work in, you can work anywhere, everywhere in drug discovery and development. So this is PDX model. On left, you probably the naked eye you cannot be able to see. On the left side is the tissue factor expression in patient-derived xenograft cervical cancer model. Today I'm going to show specifically cervical cancer because that's what the data that we recently published. And this is the preclinical cervical cancer animal model. And you can see the expression in the left-hand side. In the middle there, the graph is the primary treatment. If you don't treat with Tisotumab vedotin, there is the filled purple circles. There is no tumor growth after Tisotumab vedotin administration. On the right side is like sometimes if you give this chemotherapy, the tumor comes back. So this is that model, retreatment after paclitaxel. We treated these animals, mice, with the Tisotumab vedotin for the paclitaxel resistant model, and we still got a response with Tisotumab vedotin. What is Tisotumab vedotin? This is an antibody drug conjugate. It's if you are not aware of what I'm going to give you a little bit example. It has everything. So it has an antibody, it has a linker, it has a payload. And most of the times, a payload is a cytotoxic drug. In this molecule, the cytotoxic drug is MMAE, as a monomethyl auristatin. Basically, once the premise behind this is cancer cells, especially some cervical cancer cells, has aberrant tissue factor expression on them. So our molecule goes and binds on the tissue factor. Once it binds, it internalizes. And then once it internalizes, it releases the cytotoxic drug, and then that kills the cancer cell. That's the premise behind the Tisotumab vedotin. So Tisotumab is the part of tissue factor antibody. Vedotin is the part because of the MMAE. We are currently working this drug with Seattle Genetics, another biopharmaceutical company. So this is what our clinical trial design. You probably remember phase 1. This part is the dose escalation phase, phase 1. We had several other cancer cohorts. And then we tested from 0.3 to 2.2 milligram, different doses. And we selected 2 milligram for the dose expansion, that is phase 2. And then we tested again different cancer cohorts. However, we found some responses in cervical cancer. And then we selected cervical cancer, and then we expanded cervical cancer. And then we produced some data out of in cervical cancer. So these are the disposition. So what kind of patients that we chose? Age around 46. One thing that I would like to mention, especially Indian population, cervical cancer is the biggest killer of Indian women after breast cancer. It's purely preventable, but most of the times people, women, they don't get the cervical cancer vaccination in their early age or later. So what I'm trying to say, we're talking about the sick women with cervical cancer. You see the age here, 46. At the age of 46, they don't have anything else. They are basically at the last option is a clinical trial. So they came to a clinical trial. However, majority of these patients may not come to this clinical trial if they would have got the cervical cancer vaccination or periodical checkups with their gynecologists every year or every other year, get the pap smear done. They may not die this early. So I recommend, as a healthcare field or healthcare research scientist, I recommend you speak with your friends, family to get the cervical cancer vaccination, because it's approved in India and it's not that expensive. It's not covered by Indra Dhanush vaccine program. However, it is available in India through GSK and then Merck. So let's come back to the presentation. These are the different types of the subjects, patients who had. You can see there are previous treatment or 4, like they failed 4 prior treatments for the second line. So there may be 10 or 15 prior lines, we don't know. So we're talking about a 46-year-old on an average patient, a year-old patient might have got this disease way early in her 20s, and then got exhausted all the therapies, came to this study. So think about in that aspect, it's preventable. Cervical cancer is preventable. Think about cervical cancer vaccination. And then what we found, as I mentioned, safety, we found several things. And then you can see around 50% epistaxis. And then, however, they are grade 1. What is epistaxis? Is a nasal bleeds. So when they put their hanky in their noses, they find some blood tinge, or sometimes they has blood coming from their nose. So that's one adverse event. Fatigue, nausea, and these are very common in any cytotoxic chemotherapy. These are the common adverse events with any chemotherapy. The one thing that is not common is conjunctivitis. We found some conjunctivitis in these patients, and then what we did is, like, we gave the pre-medication with the lubricating eye drops and steroids, and before they get the and then ice pack while they are getting the Tisotumab vedotin administration. The good thing is, like, we got efficacy data, and then you see both the outside response, like, we send our scans to the separate body and they check, and then the investigators check. Both of them got the 24% overall response rate. That means 24% of the subjects, 22 to 24% of the subjects got response with the Tisotumab vedotin. And these are called waterfall plots. The waterfall plot, if you're experienced with this, the more down here below minus 30 is better. So this is a highly difficult disease to treat, and you have seen how many prior therapies they failed. but still we got 35% patients got some response. And I'll give you a case study. This is a 39-year-old patient. You can see the target abdominal lesion, huge, 35 millimeter is, we're talking about a huge abdominal lump in their, in their body. And then after treatment for, by week 13, it's completely disappeared. And then even after week 46 with this drug, it was disappeared. Again, not all patients had the same response, some, and this is one of the best response that we got. So the conclusions, basically, I would like to be quick. And TV, this Tisotumab vedotin demonstrated, very good patient manageable safety profile. At the same time, it has good efficacy too. So I'm going to stop it there. I'm sorry I took like 1.5 hours. It's all yours. Okay, can you hear me?
Yes, sir.
Okay, wonderful.
Thank you, sir. We could connect to what is there in our textbooks and also clarify, correlate the information with the matter which is there in our textbooks and what we are teaching to the students and all. So, can we take the questions, sir?
Sure, I'm ready.
There are a few questions from the YouTube streaming and the WhatsApp group, sir. One question is, for already drugs for which clinical trials are already finished, so if they are going for a drug repurposing, if they're going for repurposing, so what are the things to be considered?
So the clinical trials, there are no waivers. So if the drug is going to be repurposing, you have an advantage of going quickly, but you cannot escape anything. So I'm going to give you an example, because right now we're talking about COVID. There's this company, they have done an anticoagulant study in Ebola patients, SARS patients. So they have shown very good data. However, what they wanted to do is in COVID patient, because right now we are talking about COVID has the both endotheliopathy and then platelet coagulation as an issue. So what they have done is they have sent their application to FDA to do a study, but they still need to do another study, because even the drug is same, they are repurposing, they still need to do their phase 2 onwards. Phase 1, maybe they don't need, if they say that the drug dosing is already set, we are using the same human population. So most of the times phase 1 you can get some waivers, but not phase.
Thank you, sir. So what another question is, what is the kind of training that can particularly a postgraduate student can do if he wants to go for a clinical research field?
Yeah. I mean, it's definitely a great question. And then the one thing is like the basic, right? So the pharmacology that we learn, pharmacokinetics that we learn in our textbooks and in academics still be useful. So what you can do, I would say, try to do a CRA role if they really want to develop in clinical research. I would start with CRA. That's where you learn a lot. So if you can find these CROs, there are several CROs even in India, Paraxel, PRA, ICON, all these companies, PPD, IQVIA, they recruit freshers without any experience. They recruit, and highly recommend if somebody wants to excel in clinical research, CRA is the place to start. And I have done a couple of co-monitoring visits. they're eye openers. So what you do at sitting at a computer is totally different. What you go to the site, learn what kind of things that they do is an eye opener. So for a postgraduate student person, I would ask them to start from CRA.
There is one more question. They are saying does cosmetics. So there have been some things like Johnson and Johnson. They have changed their powder for babies, etc. So are there clinical trials for cosmetics? They're asking, sir.
There are, but it all depends like what is the purpose. So you cannot do for everything. It's definitely all big cosmetics companies, they do run their studies. For example, if it's a lipstick, they do skin permeability experiments, they do animal experiments, but not all clinical experiments. Sometimes these days a very interesting thing is happening, palatability experiments. So if a company is doing a formulation which is not a drug, but let's think about a honey syrup. So what they're doing is like a phase 1 palatability experiments. They bring healthy volunteers. They'll ask them to rate how it's tasting, whether they will go for they'll have a questionnaire. They'll fill in the questionnaire. So yes and no. Same extent, we'll see the clinical trials. Yes, there are some clinical trials. I'm assuming that even Ayurvedic companies these days, they are having their own clinical research division.
I request any of the participants from the
Yes. Anil sir, this is Ravi Kumar. Shall I ask a question, sir? Yeah. Good morning, Dr. Srinivas. I'm a teaching faculty at Sri Vishnu College of Pharmacy. Thank you for your presentation on the clinical research, which covered many areas, especially regulation aspect. I'm teaching a theory paper called Clinical Research Regulations for PG students in RA department first semester. So here we study in detail about clinical research regulations. They are very clear on each aspect theoretically, but it is a rare opportunity really to meet a scientist who involved in the practically phase 2 and phase 1, phase 3 also, you said. Yeah, actually I am able to reflect many concepts during presentation which we have studied and I hope our students also participated, felt the same thing. I want to thank you for that opportunity. So let me clarify two things in the clinical research aspect. One is about conflict of interest. We have studied theoretically about it. So being a principal investigator for many studies, can you give some practical examples of conflict of interest arises, especially for a principal investigator with a sponsor or medical doctors, etc.? How can you resolve it? Do you follow GCP guidelines when they arise? That's first.
Yeah, so so that's a good question. So let me answer that, and then we can go to the second question.
Yeah.
So I'm not a principal investigator, so just be clear. So I am at the sponsor side. So most of the times, conflict of interest happens with the investigator at the investigative side. If a PI or sub-investigator, they may have conflict of interest. I will give an example. They may have several ways. One, if I go to them as a sponsor, hey, I would like you to run this study. Are you interested? If they are already running another similar study with the same target with different company, they may say they technically have to say, sorry, we are not able to do that because we are doing this all. Second thing, with the financial conflict of interest. So they cannot. We do have 1472 forms. So you probably are well aware as per FDA. 1472 forms are basically tells you how much financial things that you can have, how many shares of our company that you can hold. if you want to run a trial with us. So it's basically between investigator or sub-investigator with the sponsors. The conflict of interest generally happens. Okay.
Okay.
Did I answer
So
your
actually,
question?
yeah, I'm following. So especially, I'm just curious to know, is there any role playing by the medical doctors which are actually in contracts to the principal investigator, because these two are the technical people about the study? Is there any possibility to arise a conflict of interest in moving forward in the studies?
Yeah, definitely. See, this is everyday thing, because that's why one needs to have, very good question, in fact. That's why one needs to have a good protocol. If the protocol is clear, what you really want to do, what kind of population that you want, what kind of statistics that you want to do, it will be nice. But the problem happens is investigator at the investigative side, he or she thinks that it's good for the patient, for the clinical aspect. Patient may get benefited. And then we, from the other side, we purely look from the protocol side. There are instances they come to us and saying that, we want to give this drug, additional drug to the patient. And then we'll say, yes, but according to protocol, it's not acceptable, because they are experts in the clinic, but they are not, we are experts in the protocol. So
right, sir. Yeah,
that's where, of course, the discussions happen all the time.
okay.
So that's why to have a good collaborating teams are essential.
So I think from your point of view, I can understand that you are sticking to protocol, right? Perfectly,
Yes.
without deviations.
Yes.
Yeah, thank you. So one more last clarification. And nowadays, most of the sponsor actually, I've seen that recent days attracting volunteers by posting study details in the websites. In case of cancer studies, you said that you're a part of many cancers. I think some cancer studies. Volunteers globally can be recruited. In this case, if are there any issues arise to comply with the informed consent process?
So it all depends. For example, if you have, let me give you an example, and I'm running study, and then you have no sites that I selected in India, and then some from India wants to participate. It's very difficult because there are no sites in India. Second thing, it's when I said very difficult, it's not impossible, because some rare diseases, the total number of patients for that disease are maybe 100. So in those conditions, there are some companies called Biomarin. They are like ultra rare disease conditions, disease. They bring the whole family, and they give the housing, they give everything for the treatment duration in different country where there are Did I answer your question?
Yes, sir, almost. But my question is that because we study and we teach also to students that participants volunteers should be from the same nation when the studies are doing at a one site. So there are again separate regulations are given specially for the foreign subjects. So in case of rare studies like a cancer, if patients are not adequate for carry out the studies at 2 and 3 level phases, then how do you overcome that challenge? In that case, if you recruit globally patients, then how do you comply with the regulatory guidelines? So that aspect you partly answered it. You can add one.
So regulatory guidelines, are you asking FDA or FDA in India?
Yeah, FDA, sir, FDA, not in India, because you're working there. No, you might have good experience with FDA.
Yes. So FDA will never directly say that you need to recruit. From this country. They definitely want population from US. If you submit something non-US, they still will, they still want you to run some studies in US.
Okay.
There are instances. One slide that I skipped. which basically answered your question. So that study was run by NIH, and it was a huge phase 3 study. And then what they did is like they tested spironolactone, and then they found discrepancies between Russian patients and then US patients. So then later they checked metabolite in their urine samples, and then they found no metabolite in 30% of the patient population in Russia, only 3% in US. So that's the biggest reason. So sometimes they look the distribution, they don't like, from one geographical location. They want more number of people.
Yes. Okay.
And again, it all depends on the which phase. If you're talking about phase 1, most of the times they don't want all over the globe. Maybe 2-3 sites is to still be fine.
Okay, sir. Yeah, it answers my question. And really, thank you once again for this opportunity, sir. It's a very clear and broad presentation, especially you touched upon so many aspects in the ethics and research regulations, which is very helpful for our students as well as for me also. Thank you once again. Really, it's a wonderful presentation.
No problem.
Any questions from the students?
Hello, sir. Good morning, sir.
Hello.
Hello, sir. Thanks for the presentation, sir. First of all, I'm Naveen from Masters in Pharma Chemistry student.
Hi, Naveen.
I have a doubt, simple doubt, sir. We have docking software, no, sir, nowadays, Autodoc, Strodinger and all. Before drug discovery, we will check we'll go for the structures and all, we'll dock it and we'll check the pharmacokinetic profiling and dynamic profiling. But still, we are, these drugs are, these structures may pass in the softwares, but not in the clinical trials. What may be the reason, sir? Up to what extent we will believe in this type of softwares and all?
Yeah, it's a good question, but again, still it's not. We're talking about complex things in living organisms. So human body, preclinical experiment, talking about several things. So at the docking side, yes, it may dock. Again, we recently had this discussion, and then I would say it's a starting point of dock and everything. You may not be able to see, everything based on docking. So you may see, okay, it's docking, it's binding, everything is good. However, it's the starting point. So based on that, you create SAR, structure activity relationship. You create different molecules and then test in large libraries in high throughput screening. Then you find something. You still need everything. There is no way. So if I say that, can we, we are multi-million, billion cells. So with the docking, you may do maybe at one planar. So 3D, 4D, several things. So I'm trying to say is like it's a valuable tool. However, it still needs biological testing.
Yes, sir. Thank you.
So if there are no more questions, thank you very much, sir. Very informative lecture, and thank you so much for so many examples, sir. We could understand easily. Thank you, sir.
I would like to add one thing that Dr. Kumar said. Yes, I do like to meet our friends, new people. I believe in one thing. This is my belief that every person is a book. Every person has a knowledge. Every person, sometimes you don't need to read the whole book, right? You just need, if I need some pharmacology doubt, I can just call Dr. Kumar, and then he will give me all the information. So I feel like every person has a lot of potential. All we need to connect through wires. That's probably, meeting people.
Pass on to our director, sir. So, Kumar, sir.
Yeah. So thank you, Dr. Gatta, for a wonderful talk. It's more than expected. You have delivered the content, and thank you for your time. And I think it's early morning, almost touches 2 a.m. for you. I don't know how many of the students realize that. So he's talking from night 11:30 to now it's 2 hours, 20 minutes. So it is 12:30, 1:30, and now 1:50 for him, almost 2 a.m. I don't know how many of us really work that much in night, and he has to catch up tomorrow. A lot of work. Yesterday also has a lot of work. So this is how we work at industry, and this is what the needs of the demand, and one need to fill in if you want to take up a responsible job or you want to do a meaningful contribution. And one point which Dr. Gatta has really mentioned, I 100% echo with him, which I'm also now practicing it and understanding it, that every person is unique and every person has potential. Though as an academic institute, we have a certain pattern of exam system where I expect that minimum is net to be met by everybody, but maximum depends with the way you like in excelling in the way in the subject or the field that you want to do should be shown in that aspect. But minimum score is something is required, but do maintain your uniqueness and find your passion and make your goal clear and be committed, be sincere, continuously work for it, and don't accept somebody says, "Want to this is so tough," and don't expect easily those things. Yeah. Make a plan, go ahead and do it. I think this is something which is best example is Srinivas itself today. I saw him very excited, anesthesiologist who wants to go for a PhD program in US. And now he's there. He got PhD and he did PA postdoc and worked with the GSK. As all of you know, GlaxoSmithKline is one of the top 3 in the world. And he worked for 10 years there. And I contributed to Meplogebar, which we're reading in the textbooks and which we're teaching as a part of treatment for asthma. It's all possible for each one of you also. So this is today's person is the example. You see, so young, how many big things he is doing, and you all also can do it. But you need to dream about it and start interacting with these experts. So we give an opportunity to all of you to ask questions and learn. And clinical research is a hot area now. There are so many jobs, especially Pharm.D students have excellent jobs. That's why we invited 5th year and 6th year students for this talk. I hardly see any question from Pharm.D this today. I think I'm glad to see that a chemistry person asked a question. And normally Pharm.D students do ask more questions, but somehow I didn't see the excitement today. But as Dr. Srinivas mentioned, not only the 2 things to learn from today's talk. One is science, another one is more than science, which is finding the uniqueness and learning from the other persons. So at this juncture, I would like to thank all the participants who have been listening for the past 2 hours and learning many things. And hope you do interact with the experts, and Dr. Srinivas will be available. If you have any questions, even in future, I can pass on to him and get his expert knowledge. So thank you, Srinivas. I am really proud of your work, I'm very glad that we are associated for some time, and looking forward for your sharing of knowledge with us and the other parts of the world too. So thank you. Thanks a lot. And have a good day.
Thank you.
Have a good sleep, and then good day.
Thank you very much, everyone. The Vishnu College of Pharmacy. Thanks, Dr. Kumar. Thanks.
Yeah, so I think we'll end the session now.
Okay, thank you. Bye.